All right, so now we're going to talk about some cell processes that are involving DNA and RNA and proteins. So we're going to start off with just kind of reminding ourselves about DNA and RNA and proteins. And we're going to do that by looking at what's called the Central Dogma. The Central Dogma of biology, or specifically of molecular biology. And what this is that it is DNA. It's basically saying that the kind of general idea of molecular biology is that DNA, and if we go through the process of what's called replication, can make copies of DNA. But if we take DNA and we go through the process called transcription, we make RNA. And then if we go through the process called translation, we make a protein. Or more specifically, we make a polypeptide. So this is our central dogma of molecular biology, is that DNA goes to RNA and goes to a protein. And this is how information flows. So remember that DNA contains all the information needed to make all the proteins in a cell. So our DNA contains all of that information. We'll look at that again specifically about how the different nucleotides are holding on to all of this information for how to make all of the proteins in the cell. It also holds all of the information for how to make those proteins, when to make those proteins, where to make those proteins. So all of the information on everything to do with the cell, proteins, how, when, where, all of those things is encoded in DNA. Now remember that RNA are small sections or small or copies of small chunks of DNA. So we take DNA, we look at some small sections, we make some copies of it, and we get RNA. So copies of small sections of DNA, and that an RNA strand, specifically mRNA, contains all the information to make one protein or one polypeptide. So specifically mRNA contains all the information to make one protein / polypeptide. Because really an mRNA molecule is going to hold on the information to make this amino acid strand. So it encodes each one of these individual amino acids that are going to be put in a row. Now this would be the polypeptide. Remember, we take our individual amino acids and then we make our polypeptide. And then we fold and turn and twist and make all these different types of bonds to make a folded complete protein. But typically, a folded complete protein is many polypeptides put together. But it doesn't have to be. Sometimes it can be an individual polypeptide chain that then is folded, and then it's complete protein. So RNA is going to contain the information to make that polypeptide or protein. And then recall that a protein is needed for various things in the cell. So needed for things like structure, for transport, for communication, enzymes. We could list many, many things proteins are needed for. They carry out all the different cell functions. So they help to make things like carbohydrates. They help to make other proteins, even. Make lipids, make DNA, make RNA. So proteins are extremely important. Remember, they're doing basically all of the cell functions. So they carry out all the cell functions. And so this is the process that we have. Remember, in our cell, we have the outer membrane there, and then we have our nucleus. And remember, the DNA is housed inside of the nucleus. So it's kind of sequestered and protected within the nucleus. And then we go through the steps of making RNA inside of the nucleus here. Then RNA is going to, once it's made, it's going to leave the nucleus and move into the cytoplasm. And we talked about how it moves into the cytoplasm because then it's going to meet up with a ribosome, and then it's going to make up this, or make this polypeptide. And then can fold up and twist and make various bonds to then end up being a completed protein. So this is our ribosome here. And then, of course, this is our DNA. And so this is our flow of information. Our flow of information goes from DNA, we make a copy, that's RNA, and then we go to a ribosome, and then we make our polypeptide chain that folds up and then is a protein. So let's take a quick look at DNA again. Remind ourselves about the structure of DNA. So remember, DNA is a double helix. And what that means is that we have two strands of complementary nucleotides, and they are held together by hydrogen bonds between the nitrogenous bases. So two strands of complementary nucleotides held together by hydrogen bonds. So remember what that meant was that we had our sugar and phosphate backbone. So we had, let's say, the sugar, and then we had the phosphate, and then the sugar, and the phosphate, and the sugar. I guess I could have the phosphate add a little bit more phosphate, attached to the sugar, attached to the phosphate, and on, and for our backbone here. So double-stranded, which means that we have one on both sides. So I'm putting my sugar phosphate backbone. This is, of course, shorthand. If we want to remember what this looks like specifically, we can go back to our structure of DNA and RNA in our biological molecules section. And then remember, when we have sticking out in the middle here is something like our cytosine and our guanine. Remember, our cytosine and guanine were held together by 3 hydrogen bonds, and then our adenosine and thymine are held together by 2 hydrogen bonds here. Of course, I'm just shorthanding with the letters as well. And remember that A always matched up with T, and C always matched up with G. Cytosine, guanine, thymine, adenine. So then recall that the information is stored in DNA in this pattern. So if we were to unzip this, meaning break this apart at the hydrogen bonds there, and if we were to just look at one side of it here, we have a code. So our code would be something like A, let's say T, 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 C, G, T, C. Let's just throw another letter on there. And so then if we were to take this piece of DNA, for example, we might say that this chunk of DNA here is a gene, which is a part of the long strand of DNA. So remember, our long strand of DNA is called a chromosome, and that we have 23 pairs of chromosomes in our body. And within the chromosome, the long strands of DNA, we can cut out chunks of that, and then we can say that those chunks are genes. Remember, a gene is a section of DNA that codes for one protein. So codes for one protein. And so if we pretend that this section here is one gene, we could say that this codes for a particular protein. We have, as humans, about 30,000 genes in our body. So on our 23 different chromosomes, we're going to have lots of sections that encode different proteins, our genes. So when we talk about our first process here, now that we've kind of reviewed DNA, the first process we're going to talk about is DNA replication. So before we get into this illustration, up here, we'll talk about DNA replication. And what is DNA replication? So DNA replication is basically making a copy of DNA. So we're making a full copy of the DNA. And this is going to be in a situation where we're going to divide the cell. So if we have a cell and we want to make another cell, then we're going to need to copy all of the information that's held within DNA. So make a copy of all the DNA so we can put one copy in the new cell and keep one copy in the original cell. And then this is going through cell division, and then it can continue doing this. So each cell can then make a copy, and then that when the cell divides, the copy can go into the new cell, and then the copy stays behind. And this is called DNA replication. So we're replicating or making copies of the DNA. So let's see, copied before the cell divides. And it must be copied without mistakes. So if we do end up making mistakes in the DNA or in DNA replication, then we can end up having what are called mutations. And that may affect the amino acids in the protein. And if it affects the amino acids in the protein, when we talk about proteins, remember, then that can affect the folding, and then if it affects the folding, then it can affect the shape of the protein, and if it affects the shape of the protein and it's not the same shape, then it won't function the same way. So DNA replication is what is called semi-conservative. And what that means is that each new DNA strand, each new DNA molecule has one new strand and one old strand. So each new DNA molecule has one new strand and one old strand. So that would look something like if we had our original DNA molecule, and say one side we're going to just color red, and the other side is yellow, so we can see the two different sides. So again, this would be our double helix. This would be, in a helical shape. Now, when we go through the process of replication, we're going to divide these. So we're going to separate them. Basically, we're going to unzip the DNA down the middle along those hydrogen bonds, and then what's going to happen is that we're going to make copies of both strands. So we'll have the red copy over here, and then we'll have the yellow copy over here, and then we make copies of both of those strands. So then the new strand on this side, we're going to use the red strand as a template to make what I'm coloring as the blue strand, and then we'll use the yellow strand as a template to make the new strand as well. And so then what we have here is that in our DNA, we now have copies. We have one DNA strand here, one DNA strand here. They both contain the exact same information, but each one has one of the old strands, so the yellow strand and the red strand, and each one has a new strand, blue strand and a blue strand. So this is what is meant by semi-conservative. So it's conserving some of the old strands, but it's semi-conservative. It's not maintaining the old strands. So the basic process of DNA replication is basically we're going to, as we kind of just described, we'll take one DNA helix, and then what we're going to do is we're going to separate it into strands. So just like we saw here with the red and the yellow, we separate the pieces, and then we're going to build a new complement for each strand. And then what we end up with are two DNA helices. And so that's what we see here, again, with the red strand and the new one, and the yellow strand and the new one. So one old strand and one new strand. So the image that I have here, down at the bottom, is basically the process of DNA replication. So what we're going to do is we're going to go through the steps of DNA replication. First, we're going to take a look at what we need for DNA replication. So the first thing that we need is helicase. Now, helicase is an enzyme, and what that enzyme does is it breaks those hydrogen bonds between the bases on the double helix. So it breaks hydrogen bonds between bases. And so this is separating the strands. Basically what I would say is this is unzipping it. And so we can actually see helicase here in the illustration. It's kind of this blue, what I think of as part of the zipper even, this blue triangle. And so if we kind of look from this point where the helicase is, so we'll kind of make a line here. If we say from this side to the right, we have our individual double helix here, our individual DNA strand. And you can see that it's even twisted at this point. So it's twisted at the first point here. We have an enzyme called topoisomerase that's going to go along and untwist it for us. And you can see here, topoisomerase is going this direction, and it is straightening out the double helix for us. Now that it's straightening out, in this area we have a nice straightened out kind of ladder of our double helix. Then we have helicase that's going to come in, and you can see that it's using its little pointy part here, and it's breaking these hydrogen bonds between the bases on the complementary strands. So we need helicase because if we're going to separate the two strands of DNA, we have to get in there, separate them so that we can then make copies, so that we can make complementary copies on each of these base pairs here. We're going to read the base pairs on each strand and make a new strand that's complementary to it. Meaning that if this is an A, for example, then what we're going to do opposite is add a T. If this is a C, for example, what we're going to do is opposite, we're going to add a G. And then we're going to build these pieces, adding piece by piece on. And we'll take a closer look at that as we move along. But the first step is we need our helicase in there that's going to go along and unzip these lines for us. Or unzip these strands, rather. All right, so first thing, helicase, that's going to go through and separate the strands for us. Now that we have the strands separated, one of the other things that we need are nucleotides. So we need those nucleotides. We need free nucleotides that are then joined to form the new strand. So here we have helicase. Helicase is separating the two strands for us, kind of going through and unzipping, breaking those hydrogen bonds. And then, in order to add complementary base pairs, what we're going to need to do is we're going to need free nucleotides to add on. So if we don't have free nucleotides, if we don't have the building blocks, then we can't build our DNA. We can't build our polymer. The third thing that we need is DNA polymerase. Now, DNA polymerase is the enzyme complex. It's actually a clump of different types of proteins, enzymes, that are going to add those complementary nucleotides to the new strand. So it's an enzyme complex that adds complementary nucleotides to the new strand. So if we look at our illustration here, we have helicase here that's going along and unzipping our DNA. Then what we have is we have DNA polymerase. We have it listed or illustrated up here, DNA polymerase. And then we have right here DNA polymerase. And DNA polymerase is going to go along. This is our DNA polymerase, kind of this yellow box here. Our DNA polymerase is going to go along. It's going to grab onto, let's pretend this is a free nucleotide here, grab onto a free nucleotide, and it's going to attach it opposite the complementary base pair. So again, if this is A, for example, it will grab a T nucleotide, and it's going to add it across from the A nucleotide. If this is a C nucleotide, then it'll grab onto the G nucleotide and place the G nucleotide opposite it. If this is a T, then it's going to grab an A nucleotide, and it's going to put the A nucleotide across from it. And so our helicase has separated the two, making those individual base pairs available. Then our DNA polymerase is going to come along and grab onto those free nucleotides, and it's going to add them opposite of the complementary. So A and T, C and G. Now DNA polymerase is very, very fast. So it's very fast and very accurate, which which is important because, again, we don't want to make any mutations. There's only about one mistake in every, like 10,000 or 10 million nucleotides that are added. Most animals would have about 100 to 1,000 errors with every replication. But it can proofread the new strand, and then it can correct mistakes as well. So DNA polymerase can proofread, and then it can correct mistakes. So as it's going very, very fast, and adding nucleotide after nucleotide as complementary base pairs, then it can go in and it can make sure that it has done the correct thing, that it can go in, it can take out any mistakes, and then it can replace it with the correct one. So we have DNA polymerase going along here, taking these free nucleotides and attaching them opposite through those hydrogen bonds. But what does that take? That takes number 4, which is lots of ATP. And so it takes a lot of energy to unwind DNA. So this topoisomerase here, this enzyme that's moving along the DNA and unwinding it to make it nice and flat. So our helicase can get in there and break those bonds. And then our DNA polymerase is going to take a lot of energy in order to take those free nucleotides and build them into a strand opposite the open strand there, the old strand. So ATP, because it takes energy to unwind the DNA, that's our topoisomerase enzyme, and to join the nucleotides. Because remember, we're building up in this case, we're building DNA, we're adding these individual building blocks and making our polymer. And so in this case, it requires energy. So if we take a look at the whole process, now that we've talked about the different things that we need for the process, we can take a look at the process as a whole and look at this illustration in a little bit more detail. So let me erase what I already have written up here, and we can take a look at the process. So what we have is, over here on the right-hand side, we have our double-stranded DNA helix, just like we would find it, in this case, in an kind of in a loose state, not all crowded up in the full chromosome shape, but kind of loose in the chromatin shape. Either way, we have our wound-up double helix here. We have our enzyme topoisomerase that's going to go along. As you can see, it's kind of this donut shape. It goes along the DNA and it unwinds it. It actually creates some nicks in there and kind of twists it around and then glues it back together. Either way, it's going to take out that twist so that we can access this nice, straight, kind of ladder shape of DNA. And that's important because then we have our helicase enzyme come in here, and our helicase is going to break these hydrogen bonds between these base pairs. So in breaking the hydrogen bonds, then we're going to end up with separated strands, 2 separate strands. Then what we have are these single-strand binding proteins, SSBP, they are for short, single-stranded binding proteins, and they're basically going to be holding open the DNA for us, kind of like fingers on hands that are kind of pulling the DNA apart, just keeping them from binding back to each other after the helicase has moved forward. So now what we have are 2 strands that are held apart and now are available. Then we have our DNA polymerase enzyme, this yellow box here. It's going to come in and it's going to start adding nucleotides. Now it doesn't actually add them directly. When we start with DNA replication, we actually have to start with an Rna Primer. So we actually start with a little chunk of RNA, and the Rna Primer is going to be attached to the DNA first, and then we're going to go through with our DNA polymerase, and it's going to read the original strand. So let's say down here we have the original strand right here. It's going to read this green base paired original strand, and as it's doing that, it's going to read each one just like we mentioned. If there's an A, then it's going to add a T. If there's a C, it'll add a G. If there's a G, it'll C. T, it'll add an A, as it goes along the DNA. So in this one, on this side, it's called the leading strand. So our DNA polymerase, we can say over here, It attached, and it's been going this direction, adding nucleotide by nucleotide by nucleotide by nucleotide by nucleotide the whole way. And then we can still see it right here. It's going to continue this direction. Now this is the leading strand because it's going the same direction as our topoisomerase, the same direction as our helicase, and so everything is moving this direction, and it can just keep following right behind helicase, just adding nucleotide by nucleotide by nucleotide as it goes. Now up here on the top we have what's called the lagging strand. And the lagging strand is built a little bit different. And the reason this is a little bit different is for something we haven't quite talked about yet. And that is that DNA runs 5' to 3'. So 5' to 3'. And our DNA polymerase can only add to the 3' end of the molecule. So what does that mean? If you look at the illustration here, you can see that there's a 5' here. This is the 5' end of this strand of DNA. And then if you follow this strand, it kind of winds around, goes along this way. This would actually end up being the 3' end on the other side. What that means is that when we look at our sugar phosphate backbone, for example, remember it looked something like this. We have our phosphate group here. This is our sugar here. On this end of our sugar, we have our hydroxyl group. And then this hydroxyl group is what's going to interact with the next phosphate group on the next nucleotide. And then we have the next sugar here, and then we have the hydroxyl group. Now, this hydroxyl group here, this is the 3' end of the nucleotide. And that's because this is considered the number 3 sugar, the 3' end. Up here, this is our number 5 sugar, and so this is the 5' end. So when we're looking at the strand of DNA and we're looking at our sugar phosphate backbone, sugar phosphate, this end is the 3', this end is the 5'. And so when we're looking at the DNA as a whole, not just one single molecule, then what we see is that there's a 5' end and a 3' end. So this, for example, would be the 5' end here, and then down here this would be the 3' end, because this is our free hydroxyl group on the 3rd carbon. So the importance here is that, again, DNA polymerase can only add another nucleotide to the 3' end, not to the 5' end that has the phosphate group on it. So when we are separating the DNA, one side can have the DNA polymerase, the leading strand, can have the DNA polymerase attach, and what's available is not the 5' end, but the 3' end. And since the 3' end is available, it can automatically add a nucleotide here. And then it scoots down, and it can add another nucleotide, because it still has an open 3' end. And then it scoots down, add another nucleotide, because it's adding continuously to the 3' end, that 3rd carbon, the hydroxyl group end. So then it's adding and adding and adding and adding in that direction. And as we said, it can continue along with the helicase, with the topoisomerase, and just keep going, because it always has a free 3' end. Now up here on the lagging strand, we don't have that same situation. So in this case, we're actually building from the helicase this direction, kind of backwards toward the left in this illustration. Now what that does is that means that when the helicase comes in, we're going to add our DNA polymerase here, let's say, for example, and we're going to add our nucleotides going in this direction, because on the other strand it's 5' and 3', so the 5' end is on the right of this illustration, the 3' end is on the left here. So our DNA polymerase is going to come in here, it's going to add to the free 3' end as it goes along, but then what it's going to do is it's going to bump into this starting point of the DNA polymerase that started before it. So if you can imagine, we have the DNA polymerase here. Let's see, I'm going to kind of draw it in red in a moment here. So here's our DNA polymerase that we're starting right behind the helicase, and it's going to go toward the left, so we're going in this direction. It's going to be adding nucleotides. Now as this helicase is moving to the right, it's unzipping more DNA. Once the helicase is, let's say, all the way to here, for example, now we have this chunk of DNA that's available for copying or for adding a new complementary base pair. Now, the DNA polymerase, remember, this guy has gone toward the left, and so now we still have this open DNA, which means that the next DNA polymerase is going to attach itself right by the helicase and work its way backwards toward the left. And then it's going to bump into where this DNA polymerase started. And so this is why this is called the lagging strand. It kind of lags behind the other strand, and that it has to piecemeal things. It has to put together little tiny chunks, keep having a DNA polymerase attach, add more nucleotides going toward the left till it bumps into the nucleotides that it already had there, and then go back and add another DNA polymerase, and then work its way along that in little tiny chunks. Now, I did mention that we have to start with an RNA primer, and we do. We have little tiny chunks of RNA primer, and this is because we have to add to a 3' end of a nucleotide. That's what DNA polymerase does. So when we unzip our DNA, we don't have any nucleotides that are complementary base paired. So DNA polymerase cannot go in there and attach a nucleotide to a 3' end because there is no nucleotide to attach to a 3' end. So we have our RNA primer that is going to be attached to the DNA first. It's one big chunk, and you can actually see up here in the top, this little section here, our DNA primase here is going to add the RNA primer. It's going to attach it, and then our DNA polymerase is going to be able to work on that RNA primer that has been added. And so then, in the Okazaki fragments, or in the lagging strand here, we have these chunks of DNA. These chunks of DNA that are going to be built until they bump up against the RNA primer that's first attached. Then we have an enzyme called DNA ligase that's going to go in, and it's going to fix these little chunks of RNA primers. So first we actually have to go in, we have to take out the RNA primer, we have to add DNA nucleotides, and then DNA ligase is going to go in and glue these chunks together. So without getting into too much detail of the DNA ligase and how that process works, just understand that we start with an RNA primer that's attached, and then our DNA polymerase can start with that RNA primer because it'll have a 3' end that the DNA polymerase can now add the nucleotides to, so that it can add to a 3' end. Then, after we've built our DNA, we've built the DNA nucleotides going this direction, we bump into this RNA primer, then we get rid of the RNA primer, and then we add in actual DNA nucleotides to that piece. So we add nucleotides for DNA in that piece, and then these things are attached and everything is fine, 5' to 3'. So our steps of DNA replication is that we have our topoisomerase here, going to unwind the DNA for us. We have our helicase come in here, break down those hydrogen bonds, kind of unzipping the DNA. Then we have our RNA primer that's going to be attached. So attach our RNA primer, and then once we have the RNA primer there, then we can have our DNA polymerase is going to attach to that RNA primer and start adding nucleotides to the free 3' end of the molecule. On our leading strand, the DNA polymerase can just keep going, following behind the topoisomerase and the helicase. In the lagging strand, we have to keep adding our DNA polymerase. It's going to be adding opposite, so it's going to go away from the helicase, and then we keep adding the next DNA polymerase that's going to be adding away from the helicase. Then we go in and we take out the RNA primers and we add back in our DNA nucleotides. Once we do that, then we have all DNA nucleotides. We reach the end of our chromosome. We've made 2 copies, and then we're done. So all of this stuff that's happening right here is actually in what's called the replication bubble. What that means is that if we have our double-stranded DNA, right, so this is one strand here, the other strand here, we have our hydrogen bonds between them, we're going to go in, and with our helicase, we start unzipping. Right, so we start unzipping, and then what we have is our DNA up here separated from our DNA down here. So we have our helicase that's going this direction, and we actually have a helicase that's also going this direction. And then in here we have our enzymes that are then adding nucleotide by nucleotide by nucleotide to build us this new strand. So we have our enzymes, DNA polymerase, that's adding nucleotide by nucleotide to build our new strand. And so then what it looks like is this is a bubble. So this is our replication bubble. So we have our replication bubble, and as we're going through DNA replication in our cells, when we're going into cellular division, we actually, what we see is we have many replication bubbles all happening at the same time. So we have a replication bubble here, replication bubble here, and then we have all of these enzymes doing their job on all of these replication bubbles. So again, then we have all these helicases that are going, that are unzipping, helicase, unzipping, helicase, unzipping, and then we have our enzymes which are adding nucleotide by nucleotide, nucleotide by nucleotide, and each of these replication bubbles. What that means is that we have a very efficient process for DNA replication. We have lots of enzymes, lots of helicases, lots of DNA polymerases, lots of topoisomerases, lots of free nucleotides, all working together to have lots of replication bubbles so that we can copy our entire chromosome, all chromosomes, very, very quickly, so that we can then move into dividing the cell, so that we can then be healing from that division, going through regular metabolism, and then continue dividing over and over again. So that summarizes DNA replication. So if we're making a copy of DNA, that's the process that we're going to go through. So now what we're going to do, we have addressed now this piece here, the DNA replication piece. Now what we're going to do is we're going to walk through this kind of central dogma. The DNA makes RNA, makes a protein. And so we're going to talk about this first step in the process, which is called transcription. So transcription is where we are going to be copying a section of DNA to make RNA. And this can be mRNA, it can be tRNA, or rRNA. So we're talking about transcription. So we're transcribing, right, which is writing something out. And I usually think of it as kind of making a photocopy or jotting something down. And when we jot something down that somebody's saying very quickly, sometimes we change things a little bit. And that's why we have the difference between DNA and RNA, which we'll get to in a moment. But we have transcription. Remember, transcription is copying a section of DNA to make RNA. And we're going to do this whenever we make tRNA, rRNA, or mRNA. And we are going to focus on mRNA because that's what's going to be the template for a protein. And when we do this, what we're doing is we're making a copy of one gene. And it happens in 3 steps. So we're going to take our DNA, we're going to open it up, and we're going to make a copy of it with and we're going to make a copy of it, meaning that copy is going to be RNA. So let's take a look at what we need for transcription. So first of all, we need our DNA template. All right, so we need the DNA that we're opening up. What we need specifically, not just the DNA, but a section of DNA. So we need the gene that will be copied. To make the RNA. So we need to have access to that DNA template. We also need nucleotides. So just like before, we need free nucleotides. Remember, the same as DNA. Right? We need these free nucleotides in order to make a copy of something, but we have U instead of T. So remember, there's no T in RNA. Instead, we have uracil. So we have the matchup between A and U, C and G. So the C and G stay the same. We also need Rna polymerase. So Rna polymerase similar to DNA polymerase, but in this case, Rna polymerase is an enzyme complex that makes RNA. That adds nucleotides to the RNA strand instead of the copying DNA strand. So Rna polymerase. And then lastly, just like we saw in replication, we need ATP because it takes energy to form the bond between the nucleotides. It takes energy to form the bond between the nucleotides. Because remember, again, we're talking about an anabolic reaction, so it takes energy. We have to put energy into the system to make those bonds. So let's take a look at the steps here. The first step is called initiation. So in initiation, what we have is a promoter section. So the promoter is actually part of the gene. Of the gene, and this is at the beginning of the gene, and this is where the proteins bind to start transcription. So proteins bind to start transcription. So if we are taking a look at our DNA, so if we have our double-stranded DNA, and then we have our nucleotides, right, and then we have our nucleotides across from them, our complementary base pairing with our hydrogen bonds holding these two strands together. Okay. So this, let's say, all of these A's and T's and C's and G's, because we're talking about DNA, right? So this is the DNA. This would be our gene, right? This section, let's say this is the start here, and this is the end. This is our gene. Within that gene, then we're going to say that a certain chunk of the nucleotides right here is the promoter region. Promoter region. And in this promoter region, this is where we have our proteins are going to bind. So we have the promoter region, and then this promoter region, not only is it where we have our proteins bind to start transcription, it also regulates when the RNA is made. So when RNA is made or not made. So the promoter region is going to be turned on or turned off, depending on whether we need it, which would be turned on, or whether we don't, which would be turned off. And then we have our proteins will bind to that. When it's turned on, our proteins can bind, and then we can make RNA, which we'll take a look at in a moment. So importantly, mutation in this area, or this promoter region, may affect if or when a protein is made. So we already talked about mutations a bit, but if we have a mutation in the promoter area, it may affect if or when a protein is made. Because if our enzymes are unable to bind to this promoter region, if there's an incorrect amino acid, then we will not have transcription. We will not be able to make the RNA. So we're going to say our enzyme attaches to the promoter region in initiation. So we're going to have initiation, meaning that this gene is turned on, meaning the promoter region is turned on. It's going to go through transcription of this gene because we want to make a protein. First we have to make an RNA from it. So then we have our attachment of our enzymes. So we have our Rna Polymerase. Is going to attach to this promoter region in initiation. Then we have step 2, which is elongation. So elongation is when we are allowing our RNA polymerase, or when our RNA polymerase is going to be going down the DNA. When it's going down the DNA, it's going to be reading nucleotide by nucleotide. So I didn't mention, but this also has to be separated, of course. So we have to go through and we have to break all these hydrogen bonds. So those hydrogen bonds are going to be broken all along here. And then we have our DNA is going to be separated a bit here. And we have this here. So we open up our DNA a bit. We have our RNA polymerase, and in elongation, we're going to be moving along the coding region. So the DNA sequence that encodes the amino acids in the protein. So we're moving along the coding region. Which the coding region is the DNA sequence that encodes the amino acids in the protein. So again, a mutation in this area can change the amino acid in the protein. If we change the amino acid in the protein, then the protein won't fold up correctly and it will not function properly. So we've opened up our DNA. We have our DNA polymerase moving along. It's going to grab on to those free nucleotides, and it's going to be adding them, nucleotide by nucleotide. So nucleotide by nucleotide moving along here. So behind it, we have our RNA being made. And what we're doing, again, is we are adding these nucleotides across. So complementary base pairing, and these are attached by hydrogen bonds, right? The DNA or the RNA polymerase knows what to attach because it can read the strand of DNA. It knows that when it sees an A, it'll add a U. When it sees a C, it'll add a G, and all of those vice versa. And so it's going to continue along and continue along until it gets to the end of the gene. Once it gets to the end of the gene, we're in step 3, and step 3 is termination. So what we actually end up with, we know it's the end of the gene because we have what's called a termination sequence. And so this is the end of the gene. This is where transcription stops, hence the termination. So our termination sequence. And this is the end of the gene, where transcription stops. So then our RNA polymerase is going to keep going along, adding nucleotide by nucleotide, nucleotide by nucleotide, and then it's going to get to this termination sequence. Once it gets to the termination sequence, our RNA polymerase is going to fall off. Once it does, then our completed mRNA strand is now out here. And then our DNA can close back up. So then, since our RNA is now out there, this is all gone. And then this DNA can then move back down and be complementary base paired once again, because we no longer need to make that protein because we've already gone through the process of making the RNA. Or it can stay open, and we can have lots and lots of RNA polymerases going along over and over and over and over again to make lots and lots of our mRNA. And then that mRNA is going to go where? To a ribosome, so that it can then make a protein. So before it does that, we have a couple of things to talk about. So this is our process of transcription. Right, so transcription is copying a section of the DNA to make RNA. We do this in 3 steps. We have initiation, which is where we have the promoter sequence, which is part of the gene, and then we have all of our proteins bind to the DNA, at that initiation location, which is called the promoter region, and we start transcription there. The process of going through and transcribing, so making complementary base pairs from the DNA to the RNA, is called elongation. So we're moving along base pair by base pair, and we are pairing them up, A's to U's, C's to G's, G's to C's, U's to A. And then as we get to the end, we know it's the end because we reach the termination sequence. Once the termination sequence is reached, then RNA polymerase is going to fall off of the strands. The mRNA is going to fall off of the strands, and then the DNA can close up, or it'll stay open so that we can keep transcribing, depending on how many mRNAs we need, depending on how many of that protein we need at the time. So recall that when we were talking about this, we have our cell, and we have the nucleus, right? So our DNA is inside the nucleus here. And this is where we go in, and now we're making, from our DNA, we have our mRNA transcript. So this is our DNA up here. And this is more specifically, we're going to say the gene that we made a copy of. And so now we have our mRNA transcript that is just the complementary base pairs with that change there with the U instead of the T. And now we have our mRNA transcript. This has all happened within the nucleus. But now what we have is what's called mRNA modification. And this is when we have our finishing touches made. So finishing touches added to the mRNA in eukaryotic cells. So remember, we're talking about eukaryotic cells in this case. We're in the DNA. We have an mRNA transcript at this point, but now we're going to do 3 things before we move into the cytosol. So remember, there are nuclear pores here, and those nuclear pores are so that our mRNA can get out into the cytosol so it can reach a ribosome and then turn it into a protein. So, what are those 3 things? So the first thing that happens is we add a 5' cap, is what it's called. And this 5' cap is basically, if we have our mRNA, it's going to be, just like DNA, 5' to 3'. Remember, the 5' is that number 5 carbon where the phosphate is attached. And then the 3' carbon is the hydroxyl group at the other end of our ribose, or our deoxyribose, depending on whether we're talking about DNA or RNA. In this case, we're talking about RNA, so our ribose. So this is our mRNA transcript. We have the 5' end and the 3' end. What we're going to do is we're going to add a 5' cap. So it's actually a series of nucleotides, of repeating nucleotides that don't have anything to do with the gene itself. And this is going to allow binding to the ribosome. It's also going to protect the information in the mRNA. So really what we add in the 5' cap is a series of nucleotides here. The series of nucleotides is what's going to attach to a ribosome, so the ribosome can recognize it, allow binding to the ribosome. But then also, if anything, in the process of this mRNA leaving and going out into the cytosol, if it encounters any type of enzyme that will eat away at nucleotides, we can start eating away at the nucleotides at the 5' cap, but it's not going to get to the important information in the gene. And even more so, the second thing that we add is our 3' poly A tail. And this is 100 plus nucleotides. A, adenine, that's why it's poly A tail. So 100 plus A nucleotides. And this, more specifically, is to keep it from degrading. And so what we do is we add a whole bunch of A's here. So 100 plus A's at the end. And then again, as I just mentioned with the 5' cap, but even more so, this poly A tail's specific purpose is so that if it encounters enzymes that start to eat away at DNA, it'll start cleaving off these adenines here, and it's not going to get to the important information in the gene. So we have our poly A tail to protect our gene information. And then the last thing that we have is splicing. What this means is that we are going to go in, so we've added a 5' cap, we've added the poly A tail, and we're also going to go in and we're actually going to cut this mRNA. And what we're going to do is we're going to cut the sections out that don't contain information for the protein that we want and remove them and glue the other pieces back together. So cut sections that don't contain information for the protein, and we remove them. So we have what are called introns and exons. So our introns are the ones that we're going to be cutting out. Introns. So our introns don't contain information, and those will be cut out. So if we go in, we cut on either side. This is going to be an intron. We're going to remove this section, and then we're going to glue this part over to this part on the other end. So if we cut out the intron, then this will be glued back together. If we cut out this intron, then this piece is going to be glued onto this piece here. So we just cut out sections and then glue the other pieces together. The other pieces are called exons. So the exons are those that have information that are for the amino acids in the protein. So they have info determining the amino acids in the protein. So this is our RNA splicing, or mRNA splicing. So we can cut out various introns, glue back together the exons, and then we have our completed mRNA. And the reason that we do this, again, we had the 5' cap, so it can attach to ribosome. We had our poly A tail, so that it does not get degraded. We don't degrade our actual mRNA. And then we cut out the introns and then glue back together the exons. And this is because we can have a particular gene code for many different proteins. It just depends on the different introns and exons that are glued together and cut out. Cut out meaning introns, and glued together meaning exons. So now that we've done this modification, now we can move from inside the nucleus up here. We have our mRNA transcript. It's gone through modification, so now we have our 5' cap and our poly A tail. We've already cut out the introns and the exons. Now this is our completed mRNA. And now it can go out through the nuclear pore, can now move into the cytosol. And now that it's in the cytosol, it can meet up with a ribosome, which is much bigger now. Either a free-floating ribosome, remember, or ribosome on the rough endoplasmic reticulum. And then we're going to go through the process of translation to turn it into individual amino acids attached to each other, which is the polypeptide. So that summarizes this part. We talked about DNA replication. Remember, DNA replication is when we want to make a copy of the cell. We're going to duplicate the cell. So we have to go through the process of making an exact copy of DNA. We don't want any mutations, so that one copy can go into the new cell, one copy can stay in the original cell. We talked about our flow of information, so going from DNA to RNA to a protein. And we talked about the first step in that, which is transcription. And we talked about how transcription requires various things like DNA template, nucleotides, Rna Polymerase, and ATP. And then it's 3 steps. It's the initiation, then elongation, then termination. And then After we go through that process, what we have is our mRNA transcript, which is still in the nucleus. Then we go through RNA modification. And then once we do that, we've added our 5' cap, 3' poly A tail. We've gone through splicing. Then we can leave the nucleus and go out into the cytosol and meet up with a ribosome. And that's what we're going to talk about in the next one, is we're going to continue on with the central dogma, now that we've talked about transcription. Now we have our RNA molecule, specifically our mRNA molecule, and next we're going to talk about translation.